the fastest 4 hours that you've ever sat through. So let me go ahead and share my screen. Okay, so yeah, so I think this is what Dana was referring to, and it is downhill from here. This is the coolest thing in the talk. But my work has primarily been centered around when I started was really human focused and structural pest management, and now I've sort of moved into primarily animal health and the vectors, insect vectors associated with animal health. But most recently, I'll go into this just a smidge, or Poosh now is a human virus that is concerning, that is transmitted by Aculocoides, which is the group that I study. So this is just a representation of what Dana just went over, but I just wanted to highlight the variety of insects that I've worked with over my career, from termites, fleas, bed bugs, ants, to filth flies. But I've always had an interest in Aculocoides. I've worked with Aculocoides biting midges now for about 10 or 12 years. And I started more on the taxonomy side, and it sort of just progressed into now I do full-blown genomics, transcriptomics, a lot more of that side of it. And I'm very, very sorry to start the talk off this way, but I do need to make sure that everybody's on the same page. I know we've all learned about alleles in our genetics class, but let's just get a quick refresher. So I think we're all familiar with alleles that actually have a phenotypic change in an organism, either with the homozygote, heterozygote, and homozygote recessive, like eye color, right? And so depending on the alleles that your parents possess is going to dictate what color eyes you have, or a variety of other traits. But primarily in population genetics, we're concerned with what's considered we've termed neutral alleles that sort of are just in the genome and don't actually make a phenotypic response, and that way we are able to monitor their movement throughout populations, but they're not being selected for. And so it's a really nice way to look at things like ancestry, invasion history, or movement of different populations. And primarily when we look at the alleles that we're actually looking at, is you'll see primarily two different types. And so today I'll talk about SNPs. And so SNPs, it stands for single nucleotide polymorphisms. And what this is it's really is just one base pair change at the same base pair across a gene region. And depending on how many, right, if you have an A or a G there in a population, that denotes the heterozygosity, or if you have if it's fixed for A in that population, there's a lot of homozygosity. So we can determine those levels based on that one single nucleotide polymorphism. I'm also going to talk a little bit today about microsatellites. So microsatellites, it's actually a little easier to tell sort of the homozygosity and heterozygosity with microsatellites. So this marker right here is a homozygote. So both parents have the same allele. And this would be a heterozygote. But microsatellites are long repeat regions within the genome. So you'll see these motifs. Here it's a GTC motif, and it's just repeated. And the number of repeats actually change depending on the population you're in. And so the size of those alleles change. And that's sort of how we measure the change in alleles with using microsatellites. And so we do a lot of different things with alleles, actually, from ancestry. I don't know who all has taken a 23andMe test, but that's all using alleles. They have a set of alleles that they compare to the set of alleles from different regions around the world, and that's how they come up with the percentages of this percentage or that percentage. You can also sort of denote which species are what based on who's sharing genes and what alleles show up in these different populations. My wife actually works for a zoo, and so allelic diversity is really important in conservation. I think we're all familiar with the cheetah examples. Cheetahs went through a huge bottleneck, and it really reduced their genetic diversity. And so, in an effort to bring certain species back from these brinks, we need to make sure that we increase genetic diversity in our pairings and things like that. And so there is absolutely a little zoo tender database where they'll match up the uh, individuals that are the least likely to have a lot of inbred genes and things like that for conservation efforts. And then finally, I think one of the most famous ways in which we use microsatellites in particular is for forensics. So if you murder someone, how they will determine that you murdered that person is with microsatellite markers. And I think the set that they're currently using, it's the chances that you share that same exact microsatellite marker set with another person is like 1 in, I don't know, 400 billion or something. It's some crazy amount. Now, my primary focus has always been population genetics. And so what we do is you have the entire gene pool exists, right? The total allelic diversity exists within a population. But how those alleles are structured within sort of subpopulations gives us an idea of how each population is structured. It can also allow you to figure out the invasion history of certain invasive species. You can look at gene flow amongst populations. And so if you're trying to determine whether or not you have multiple species or not, this can be a really powerful tool. And then you can look at the population changes over time using these alleles. So that's just a quick brief introduction about population genetics. And now we'll sort of get familiarized with Culicoides biting midges. This is a really cool group to study. I am biased, of course, but there are about 1,400 species known worldwide. The larvae, and primarily we use these different wing patterns to tell the species apart. There are, I can't remember the last time I checked, I think there are about 40 different subgenera, and each subgenera has its own sort of unique wing pattern. And so it can be easy-ish to identify them, but we'll talk about why it's a little hard to work with this group here in a second. The larvae are semi-aquatic, and depending on what subgenus they're in, dictates what larval habitat they're in. So I say that they're semi-aquatic because they hang out in the margins between sort of the dry substrate and the wet substrate. So they like that in between, that muddiness. You can see, so the where we would collect the larvae and the pupae would be sort of right around here. The majority of them do like stagnant water, and they'll just breed along the pond margins. But then there are a lot of groups that have sort of specialized in the different habitats, like fresh manure, semi-fresh manure, rotting cactus. And I think there's even a group that somebody described it as they found a larvae living between the moisture that was contained between two leaves. So there's a lot of diversity in the larval habitat as well. And then most females will take a vertebrate blood meal, which is why most people end up caring about this group, because that life history trait makes them really good pathogen vectors. There are a lot of specialized species that will feed primarily on either large mammals, small mammals, birds. Then there are some generalists. And yes, there are even reports of Culicoides feeding on fish. So when the mud skippers come out, then they'll get attacked by mosquitoes and Culicoides and all types of stuff. So nothing's safe. And then there's also one really cool group, just to sort of highlight the uniqueness of this genus, that doesn't even need to take a blood meal at all. It just steals it from the mosquito that does all the hard work. So there's a group in Southeast Asia that will feed on the blood meal of Anopheles mosquitoes, and it will just take it straight from their abdomen. So like I said, I love this group. It's really cool. but it is incredibly hard to work with. And the reason is because they're super tiny. So this is a picture of a culicoides of average size next to a giant mosquito. And so any behavioral assay, any dissection, getting genomic data, anything that you do is amplified in its difficulty because of how incredibly tiny they are. And so I think that's why they've sort of lagged behind in the available resources. But again, they still are a really important pathogen vector. And so it's important that we continue to work on these things. So these are Culisomorpha. They share a common ancestor with all the other blood-feeding Nematosfera. So our black flies, sandflies, mosquitoes. They're also a pretty old group, about 195 to 100 million years old. This specimen right here, I think, is in New Jersey, Amber, something like that. I think so. This is maybe 60, 65 million years old. And then this specimen was collected, 5 or 6 years ago. And so you can see the body plan hasn't really changed all that much. They've found their niche and they've gone to it. Now, they have had a ton of time to evolve, and yet, which I think is plenty of time for them to have finished, but of course they're not finished. And so what we end up seeing is actually a lot of species complexes that arise. And oftentimes this happens in our vector groups, which will complicate gathering epidemiological data, species distribution data, things like that. And I actually will talk a little bit about the Varipennis group today, but almost every major vector group within Culicoides has at least one or if not a couple species complexes or within them. So I mentioned that just their feeding behavior makes them pretty good pathogen vectors. And so there are a number of viruses, protozoans, and filarial worms that are transmitted by this group. They can also, but unlike mosquitoes, they don't have that antiseptic when they bite. So it can actually be really painful. I don't know if anybody's ever been bitten by a Culicoides, but it hurts a little bit. And it can also cause what's called sweet itch in horses. And you can see that an example here. It's really irritating for them. And then there's some major viruses that are transmitted by Culicoides. African horse sickness is one of them. That had a major outbreak in Southeast Asia recently. That was newly introduced. Oropouche, like I mentioned, is newly moving northward and is threatening to be introduced into North America. Blue tongue and EHD are probably the most well-known, and they run rampant and affect mostly sheep and deer. Here in North America, deer are the primary primarily affected livestock, and in Europe, it's sheep. I think it was 2012 or 20, yeah, I think it was the 2012 outbreak. Spain lost 30% of their sheep to blue tongue. So these could be incredibly economically devastating viruses. And then vesicular stomatitis virus is another one. We just recently had an outbreak where we'd never had an outbreak before in Southern California, and it actually got into some species that had never been reported to be susceptible for. It actually got into some rhinoceroses at a wildlife park. So, like I said, I know they're tiny, but it's a really important group to sort of make sure we stay on top of. So from a population genetics standpoint, what I bring to our unit is dedicated to working on every aspect of this. And we have virologists, we have climate scientists, we have behavioral people, we have ecologists. And so I fill more of the population genetics role and genetics in general role for the insects. But there's a lot of overlap between data that epidemiology needs and data that population genetics can provide. And so in one of those is determining the invasion history of an invasive population. And so these, are really common and devastating invasive species. But if this happens to a vector, it can have, instead of having uh, or a livestock vector, it can bring new pathogens with them as well, into areas where they weren't. And so it becomes important to make sure that we know exactly where these invasive species could be potentially coming from, so we can stem that, cut it off at the source as a potential way of mitigating the introduction of these harmful pathogens. And so I do have one project working with culicoides and invasion history. And so just, isn't this super pretty? Yeah. I didn't get to go here, so don't hate me, okay? But this is a little atoll actually right north of French Polynesia. And yeah, it's tropical paradise and everything, except for when there's like 100,000 biting midges that all come out, and there's like a 2-week period where they have to actually shut down this whole island and the resort because the biting midge pressure is so bad. And so what this French Polynesia, American Samoa, these South Pacific islands wanted to know was, are they moving midges between each other, and would it potentially be feasible to, if they eradicated them from these islands, are they going to just get reinfested from a different island? And so we ended up looking at the population genetic structure of each one of these islands. And so we're still working through some of this data, but some of the preliminary stuff that we've seen is that within this little, I say this little island chain, this is like 2,000 kilometers difference, right? So this is the other thing that when we're working with culicoides, we sort of assume that they're not very good dispersers, except for they're incredibly good dispersers, and they can get picked up if you move sand, you can move them. This species in particular, there's also a chance for on small islands that wind can blow them to each other, and that's actually what we see is it looks like there are patterns of humans moving them between sort of these bigger islands, so between like Fiji and Samoa, Cook and French Polynesia. That's more than likely human assisted movement. But then islands that are sort of less than 200 kilometers apart, there's a heavy movement between those, which sort of indicates that they're potentially moving themselves via wind transmission. And there are sort of 3 distinct populations. We see Fiji has its own, Samoa has its own, but then you get over into this Cook French Polynesia group, and there's a lot more gene flow amongst that. And so the short answer for them is, yeah, if they eradicate them, there's still gene flow going on. And so no matter how much they sort of, if they do try these eradication programs, there's a chance that they're just going to get reintroduced from potentially one of these other islands. So next, I kind of want to talk about gene flow in particular. So I mentioned that some of these vector groups have species complexes. And the reason that they're in a complex is because we can't morphologically tell them apart. That's really the only reason. But the genes will tell you. They can tell each other apart. If they are different species, for the most part, right? I'm generalizing here, but they will tell you, they can tell each other apart, and the alleles within these populations, within these species, will tell you who is and isn't the vector species. And so that's sort of the approach that I took when looking at this group. And I'll give you a practical example of why this kind of is important. So when you're monitoring a vector population, if you have, let's say you test 30 individuals and 5 of them come up as positive or infected for whatever pathogen, you have a certain infection rate, all right? But if there's a cryptic species back there that you actually can't differentiate, and in reality, you only had 15 of the vectors, that you called 30, you're effectively halving your estimated infection rate. And so all of your epidemiological data isn't as accurate as it should be. And so this is more than likely the case that's been happening within the Culicoides varipennis complex for a long time. And so when it was originally described in the early 1900s, it was just Culicoides varipennis. That was it. But its distribution was like Mexico to British Columbia to like Maine. It was the entirety of the United States, or the entirety of North America almost. Then in 1957, a guy came along and started describing all of the variation that he was seeing in these populations. And he eventually coined the Chilocoides varipennis complex and denoted 5 subspecies. Well, then in the year 2000, they introduced some molecular tools to be able to look at these different species, but it was isozymes. So I'm not sure if anybody's familiar with isozymes, but that's the precursor to the precursor of microsatellites, I think is essentially what. So it's not the most refined tool, but it was the first time that it had been looked at genetically. And what their conclusion was is that there were actually only 3 species. So they took those 5 subspecies and made them 3. And so this Chilocoides sonorensis here, this is our vector species. That's really the one that everybody kind of cares about. And so once they use those isozymes to determine that there were only 3 species, they then were going to barcode them and determine, we're like, okay, great, now we can just barcode it. We'll pull out sonorensis from everybody else. Well, it turns out that they couldn't do that. They were having trouble. Sonorensis and varipennis were never separating out using CO1. And they actually tried a bunch of mitochondrial genes and never could really get that separation. And they all sort of chalked it up to misidentifications and things like that. And then that's kind of how they left it. So I came along and started doing my PhD, and I wanted to re-examine this group with some more the advanced molecular tools that we had at our disposal. So what I did, the aim was to determine the exact number of species within the varipennis complex, and then look for any population structure within the species that we actually found. And then potentially identify any gene flow between specifically sonorensis and any of the others, because we know that some of the species live in symmetry. And so if you've got a vector species hybridizing with a non-vector species, you have the potential for moving those genes into a non-vector and potentially causing problem. So those were my 3 sort of main goals. So what I did was I took, this is the map from Jones's 1957 work, and this is the distribution that he came up with for those 5 subspecies. And so I went and collected midges from these 13, 17, I don't know, I forget now, but they were from these sites here. And I tried to cover both sympatric and allopatric populations of each of the subspecies. And then so I collected all of those midges. I sent off 8 to 12 individuals per population. And I didn't morphologically identify any of these other than what was already available. So some of them are easier than others to see that morphological variation. So I noted those, but I was more interested to see what the genetics was going to tell us. So I sent these off for dd-rad sequencing. And so what dd-rad sequencing does is it cuts at certain specific areas across all populations. So instead of doing a shotgun approach and sequencing randomly, it's targeting certain areas so that you can ensure that you get those SNPs. You can get a lot of SNPs across all of your samples. And so after I did that, I was able to find about 3,600 SNPs. And the way that we denote different populations, so in this case, my populations are actually going to be different species, is with a K. So the best K from the data that I input after my analysis was K = 5, which I was really excited about because I was like, oh, K = 5. That's the 5 subspecies that Wirth had originally seen. Except for the fact that you'll notice that Chilocoides australis drops off of this map because I didn't find any molecular evidence for australis. That doesn't mean it doesn't exist. It just means I didn't find it. What I found instead was this unique cluster over here in San Diego, which I have done a lot more work on, and it turns out that that is actually a new species that we didn't know about before. And Worth actually in his original work said, hey, there's a weird group down in San Diego, but I'm not going to call it a new subspecies because he just said he couldn't find the morphological characters to justify it, but he said it is kind of weird. So turns out Worth was right on pretty much all accounts. So we do have at least 5 species within this complex. And you'll see that the original subspecies designations pretty much matches the distributions of what the SNPs are saying are the true species as well. So this was really, really encouraging to see. And then you also see that there is a lot of symmetry between the groups. So Sonorensis is definitely living with Albertensis. It's also living with Varipennis. So there's and Occidentalis. So the main vector species is living with pretty much every single of the other species, the members of this complex. And so that scenario in which we talked about monitoring and testing these vector populations has definitely been a case. I think we've been underestimating the overall infection rates because we haven't been accounting for Albertensis at all. So we just throw anything that kind of looks like Sonorensis in, and then we'll test it for virus. But there's likely Albertensis is likely not a vector, and so we've probably been underestimating the viral prevalence in some of these areas. So we've also I went through and I did a lot of different clustering analyses, and they all sort of say the same thing, that these are true, unique individual species. But one cool thing was there were 6 or 7 putative hybrids that were found. And so this is just individuals that were in structure found to have ancestry from 2 different lineages. And so you can see them highlighted here. So we've got an Albertensis and a Varipennis cross, a Varipennis and a Sonorensis cross, and an Occidentalis, an Occidentalis and a Sonorensis cross. And so we wanted to, there's a way in which you can tell the direction of these putative hybrids, and it's through looking at the mitochondrial genes. So that was the other thing, is we thought that we had the answer now. We have the SNP IDs on these species. Now we can just barcode the species, the individuals that we have the SNP IDs for, and we'll solve everyone's problems. We'll get the barcode for Sonorensis, and it'll be easy peasy. And we'll solve. I was very, I was very optimistic about what was going to happen. But the other thing that we could do with sequencing the mitochondrial genes of these individuals was determine directionality of these hybrids. And so I had a hypothesis in that these species end up having, they have different larval habitats and different mating sites. And so you see that Occidentalis will actually, it lives in these high saline environments, and it mates back at those environments, whereas Sonorensis mates at the host. And so the females of Occidentalis are going to be accosted by Sonorensis males at the cow, but the reversal never happened. The Sonorensis males never go to the breeding sites of the Occidentalis. So you'll never see a hybrid with a Sonorensis. The prediction was you'll never see a hybrid with a Sonorensis female laying in these types of habitats. And the same thing should be true for the Varipennis as well. And so that's kind of what we're predicting is all of the hybrids we should see should have Occidentalis's mitochondrial. So they should group with the Occidentalis haplotypes. And then we ended up actually seeing the data, and it surprised us all. So we were correct in that these two hybrids, now granted, it's only an N of two, but the hybrids did have, of the occidentalis and the sonorensis, did have the um, occidentalis maternal haplotype. So that scenario does end up seeing, it looks like that is correct. So the sonorensis males are mating with occidentalis females at the host, but they're never going to meet an oxid, or a sonorensis female is never going to see a occidentalis male at the host. So this looks like it's going to be one. If the hybrids do happen, it's going to be in one direction. And then you'll notice that there's this split here. So there's a chance that some of the or the Northern California occidentalis are cut off from the rest of the other populations of occidentalis that we saw. And this is probably because of how unique their larval habitat is. So when we look at the population structure, we also see the same sort of thing. Each individual population that we looked at had its own population that we didn't see in any of the other species. All the other species across all the areas that we sampled were 1 K equals 1. Meaning that there was gene flow from Texas to California to Idaho for sonorensis. It was one big panmictic population. But occidentalis absolutely does have population structure, and it likely is because of their unique larval habitat. The other thing that you'll notice is this huge haplogroup right here, consisting of sonorensis, varipinus, and albertensis. And so what ends up happening, here is the reason that nobody's been able to barcode these species is because they're sharing a mitochondrial haplotype. And this is not just at the CO1 gene. I've since sequenced the whole metagenome for all of these subspecies, and it's identical across these 3 species. So that divergence has either happened so recently that it hasn't given enough time for differentiation, or that group has just found a haplotype that a mitochondrial haplotype that's advantageous for all of them, and they sort of just share it. But the long story short is there is no way to barcode them using mitochondrial genes to tell sonorensis, the vector, apart from the rest of them. So what I ended up doing was taxonomically breaking the varipinus complex up from the 3 species that it was, and I made it 6 species. So the 5 original subspecies, I elevated everybody to their species level, and then described that new species from Southern California. But again, like I just mentioned, the inability to pull sonorensis out from everyone else was a hindrance for vector surveillance efforts. I mean, differentiating sonorensis from varipinus occidentalis was kind of hard. Differentiating it from the new species that I just described is even harder. And so in an attempt to make sure that the entire Culicoides community didn't hate me or tar and feather me, I set out to actually make a diagnostic test to be able to tell these species apart. And what I started using was microsatellites. And so the RADseq technique that we used to develop the SNPs, it's actually really, you will never get the same SNPs. I could give you the same samples that I used, and you could submit it for sequencing, but you would never get the same exact alleles that I did. You would likely get the same results that I did, but the reproducibility is, it's not there. And it's also really, really expensive. So microsatellites offer a way to actually make reproducible data, and microsatellite markers are like $7 to order. So they're a much more cost-effective way of separating these out. And so what we did was we used a bioinformatics program with the old sonorensis genome to comb it for potential microsatellite markers within the genome, and it found quite a few, and so we tested them and came up with a 21 marker data set that absolutely still it does separate the species out into the five species that we found using our SNP data, and structure clearly differentiates them into those five species. So we were able to do it. But 21 markers is a lot to ask anyone to do for like a single species identification. So we employed a machine learning algorithm to see if we could improve this data set. And so we used it was a random forest, and essentially what this did was it took all of our markers and sorted them from sort of the most useful in species delimitation and less useful. And so we kind of have we have this cutoff from a 7 marker and a 4 marker data set. So this 4 marker data set uses the 4 most useful and the 7 most useful, right? And then we tested those against our SNP data and our 21 marker set. And what we found was that the 7 marker data set performed really well, but the 4 marker data set performed well as well, almost as well. The confidence, just if I was going to put a confidence interval on it, the species delimitation for the 4 marker data set was like 93, 94%, but it was like 97, 98% with the 7 marker data set. And these can actually be multiplexed. So it is just a single PCR, but then you do have to run them on a sequencer to be able to get the alleles and then call the what is it allele, and then you have to do so there is quite a bit of analysis that goes along with this. And so we still weren't really happy with this for its usefulness for other researchers, but luckily what we found was one marker that was specifically amplified in only sonorensis. So remember, sonorensis is our vector species. So it doesn't matter for the majority of researchers, they don't necessarily want to find the other ones. But if there was an assay that said, yes, you have sonorensis, no, you don't have sonorensis, that's a lot more useful. So we found one marker that preferentially amplified in sonorensis and did not amplify in any of the other species that we tested. And so you can see here, this is, I think, 11 or 12 populations that we tested this. So this is a single PCR using one set of primers, and then you gel it, and either you have a band, and yes, you have sonorensis, or you don't. And you can crush up a bunch of individuals in a pool of individuals and tell this or not. So we were really happy with how this came out, and sort of this is like the culmination of what I started off with was, let's tell sonorensis apart from everybody else, and then also here's a usable assay to help improve vector surveillance efforts. If you want to know, if you're in one of those areas where it's really hard to tell the difference between all these species, here's the molecular marker to be able to aid you in that. And so just to finish up with culicoides, some of our future work, we're really getting into, like I said, or like Dana mentioned, getting a lot more into the genomic side, the transcriptomic side. So they've got a project looking at gene evolution of certain genes. Right now, we're focusing on the odorant receptor genes, and we'll be testing a lot of stressors on culicoides to look at their gene expression profiles. We also redid the genome. There was a genome, but it wasn't as usable as we would like it to have been. It was in about, I think the original was like 8,000 scaffolds. They got it down to about 4,000 scaffolds, but it was definitely incomplete. We had some transcripts that didn't map back to it. And then they also had a pretty big problem with gene duplication. And so this is what the old genome looks like. This is the new genome. We just finished our running that. So, and you can see just how much more complete it is. I think their longest scaffold was 766,000 base pairs. Our longest scaffold is 48 million, which is an entire chromosome. And so we also did some high C mapping, which was really beneficial and into getting the structure. So Culicoides only have 3 chromosomes, but you can see them very clearly here in the high C data. It was clear for me. If you don't know, if you never looked at high C data, I think it probably is pretty difficult. But so yeah, that's sort of our future work with Culicoides. Now we'll not to give anybody whiplash, but let's talk about insects as food. So this idea kind of came not from, people have been talking about eating insects, and people do eat insects all over the world. But this idea of using insects in livestock feed has really become a lot more, for lack of a pun intended, palatable for people, right? They don't want to talk about eating insects, but they'll feed them to the things that they eat, no problem. And so when I started my postdoc, Lee and I sat down and were thinking that, there's all of these pest flies are already being produced in these agricultural facilities. And so if there was a way to harvest those flies and then utilize them back into the feed that we use for other agricultural species, you form this sort of circular agriculture. And it was an idea that gained a lot of traction within the USDA and allowed me to do some of my postdoc work. And so just to show you that this is actually being implemented, here's a new dog food that came out, and you'll see that one of the main ingredients here is black soldier fly larvae. Now these are farmed, but our work is more focused on potentially utilizing wild collected flies as a means to supplement protein within livestock feed. And so this paper just came out, and it was done by our we participate in a program called the VRSP program. It takes veterinary students in their first or second year, and over the summer, instead of taking a break like they should be doing, because vet school is hard, they end up just doing 10 or 12 weeks of research instead. So Courtney Van Nest and Sabrina Swistek worked on this project and are the main authors of this paper. And what we did was we collected flies using a new mass trap that Lee and I sort of built and have been testing. And then we screened those flies for pathogens and tested a couple of ways of disinfecting these flies to then potentially use them in the feed, right? So a food safety aspect. And we also wanted to make sure that our disinfection techniques didn't destroy the nutritional quality of those flies that we were using. So just to give you an idea of what this mass trap looks like, this is, I think, version 5. I don't know, Mark 5. Lee's had so many of these. I think I came in at Mark 3 and developed them. This one looks a lot nicer than some of the ones that we've been using. But essentially, it uses a car radiator fan. It's got a lot of electronics on it. We've got a timer, so it turns on and off. And this fan will suck all the flies in to this netted area, and then the funnel will push them into this car garbage can. And so from our first year of using this trap, so this is the actual fly version of it. The other one was a mosquito version. We tested to see if the number of flies that we're removing from the environment actually affected the population of flies. And it turns out, so the dark bars are the biomass that we collected. And so we get this huge uptick, and then sort of it stagnates over time, over the 14 weeks that we were trapping. You can see that the lighter bars are the overall fly population on that facility, and you can see that we didn't do anything to them. Which we were hoping that we could have reduced the fly population, but we didn't. But silver lining, that means that there is a sustainable just source of protein on these facilities that you can just tap into and harvest as many flies as you can. So here's just the pile of dead flies that we collected sort of per week. And actually this last year we went at it again, and we did much better. I think we collected like 1.5 kilograms of flies our first year, and we went up to 12 kilograms of flies our next year. So this is a trash can. This is a 44-gallon trash can just full of flies that we harvested. And the reason that we harvested them, this is just to give you an idea of the density of flies that exist on these facilities. Just wait for it to kind of speed up, but you can sort of see all of the flies that exist, and the fly pressure on some of these ag farms. It just gets so high. So real quick, what we did when we took the flies, we collected flies from different facilities. So we looked at a dairy and a poultry farm, and we did it over 3 different months. What we wanted to see is the bacterial community over time and then across different facilities, because our idea was you could potentially take flies from, let's say, a dairy and then use them in a poultry, because potentially the pathogenic bacteria that's on one might not affect the other, and vice versa. And what we did find was that the bacterial community it varied on farm by month. So depending on when you collect, you will get different bacterial diversity. But there is still quite a bit of overlap of pathogens or potential pathogens that exist no matter what. And all of these are associated with feces and things like that. So Proteus mirabilis and things like that. So no matter what, for food safety purposes, I think a disinfection method is going to have to be implemented. And so what we tested originally was we did just a drying. We did a drying at about 200 pretty Fahrenheit, yeah, and for 1 hour. And then we milled the flies up after it to turn them into sort of a powder. And then a second batch we heated at like 400 for, I think it was 2 or 3 minutes. And just to sort of flash heat anything after they had already been milled. So to potentially expose sort of the internal bacteria and things like that. And actually what we found was that the milling process itself significantly reduced the amount of culturable bacteria in these. And in some cases, the extra heat treatment did increase or decrease the bacterial growth, but in other cases it didn't. And so to fully get rid of the bacteria, we're looking at other sort of disinfection techniques, but this is sort of our first kick at the can, if you will. And so because we did apply such a high heat, we really wanted to make sure that we didn't destroy the protein content, because that is one of the biggest sort of benefits of using these flies is they have about 70% protein, which is really, really high. And so when we did the heat treatment, we did the milling, we still maintained a really, really high crude protein content even after our disinfection. And so then we also looked at the amino acid profiles and the other nutritional value of these flies to compare it to other harvested insects and colony collected or colony raised house flies. And it all sort of looks the exact same. So it does look promising for feed, but I think the one hurdle that we need to overcome is definitely the food safety aspect. And so that sort of, with this work in particular, we are going to continue working on disinfection. There's a scientist here in our facility. He's in the grain quality side, but his entire background has been food safety all his career. And so he has some ideas on ways we can test and try to make these flies a lot more safe to incorporate into a diet. We're also going to look at digestibility of the flies, because there is still a lot of chitin and things like that that we have to account for. There's also potentially a project that we're going to work on to utilize these as a fertilizer. And then we also want to measure the CO2 emissions that come from feeding these a fly-based diet to livestock to potentially see if it reduces carbon emission as well. So I'm going to plug my lab really quick. I have a ton of sequencing capabilities. So if anybody's interested, I have Sanger sequencing that does short Sanger sequences, but it also does microsatellite analysis. I have a P2 Solo, which is the long read technologies, and then I also have an Aviti, which, if nobody's familiar with this is sort of the next generation short read sequencing. It uses a hi-fi short read sequencing, so this is essentially what PacBio has moved towards for their long read sequencing to increase their quality, but they've done it in a short read sequencer now, and it produces really, really wonderful data. And so with that, I'd like to thank all of the funding sources. Primarily, it's all USDA and has been my whole career. I thank the unit, the Abinger unit here at in Manhattan, and a list of collaborators, which it should be a mile long. But these are the people that help me out the most. And then I have some university collaborators that I couldn't have done this work without. And with that, I'll take any questions.
Awesome. Thank you, Phil. Okay. I was writing furiously while you were talking, so I have tons of questions, but I'll leave it open to everybody else first since we'll talk
Yeah,
afterwards.
I think there's a hand raised.
Yeah, I'm trying to get my Brady Bunch view so I can look at the hands up. Everybody just chime in. You can start talking, whoever's hands are raised here.
Are you aware of the CEIF? I'm pretty sure you would be, right?
The CEI,
CEIF, Center for Environmental Sustainability through Insect Farming? Or is it because you're specifically working with like the pest ones that you don't like farm them at all?
oh yeah, no. I mean, like I know that there's a ton of work that's done. That's primarily what's done, right? Is the farmed ones. But this is potentially just tapping into the, all of the pests that we have on these ag facilities. But there's a bunch of groups within entomology. There's a special collection that just came out about insects as feed and things like that. And so I'm not familiar with that group in particular, but yeah, no, I know of the efforts to farm raise them.
Alex.
Hey, hi, I'm Alex. I'm studying entomophagy, and I have a couple of questions.
Yeah, absolutely.
Okay, to begin with, is there any way you could possibly collect them in their larval stage in those numbers? Do you think there's any way that could happen?
I don't know. Maybe. So they're breeding in the manure, right? And the other place that we're seeing a lot of these the maggots is the feed. So if the feed's not properly stored or swept up or things like that, and this facility that we're actually working on, what they do is they take all the feed, they put it in a barrel, and they dump it on the soybean field next to them. And so if you go into the soybean field, you can sort of just kick the soil over, and there's maggots sort of underneath them. I think it would be a lot more labor intensive to do something like that. And it would be harder for the farmers to buy in. So the mass trap is a way in which they sort of just plug it in and passively harvest the flies. But if you have ideas, I'm all for them. Yeah, absolutely.
Yeah, sorry, I just rear them. I wouldn't know how to trap them
Sure.
that. Yeah, the
But
same
no,
it seems the larvae of holometabolous insects tend to have more nutrients than the adults, so that's the
yeah,
eating
exactly.
stage.
Yeah, no,
Yeah,
and
aquatic insects generally have more balanced fatty acid ratios than terrestrial insects.
right. And this is the thing that we sort of why we want to compare it and see if it's even viable to, right? Because you can obviously farm them, and the larvae are what most people use and utilize some. So I think there might be even some pupae that people use for like maybe some of the beetles. But it's because it's the chitin too, right? It's hard to digest.
Yeah, larvae have much less chitin than adults too.
Yeah. And so I don't think it's ever wild collected insects is never gonna replace farmed insects or the larval, but it as like a supplemental or something like that.
Yeah, it's definitely a cool idea.
Yeah, I think that's what we're thinking. So thanks.
Okay, yeah, probably just my other question is like, what methods did you use to determine the nutrients, like the crude protein? What did you use for that?
Yeah, we send it off to a company, and All of that, all of those methods are definitely in that paper.
Oh,
But
okay.
yeah, no,
Okay.
we paid the company to do all the nutritional analysis. But the actual test that they use to determine is they gave us all that information and everything. So I just don't remember off the top of my head.
Okay, great. Thank you.
Mhm.
So actually, would you mind stop sharing your screen, because
Oh,
that way you can
sure.
see everybody. It'll be easier
Yeah,
to
absolutely. There you go.
see. So, I have a question about, you mentioned this cool little machine, and I missed the name. And
Oh, the Aviti.
what do you call it again?
It's Element Bioscience is the company that makes it, and the Aviti, Aviti.
Okay, got it. I can do the rest. So
Yeah.
thank you, because I am looking for something, and it sounds like that's short read. So
Yeah, and it's awesome. I couldn't recommend it more.
okay, all right. So I have another question. One, I find it fascinating you didn't mention to these crowds that culicoides are the only ones that pollinate cacao.
Yeah, so it's actually a Forsythia. It's a ceratopogonid, but
Right.
it's a closely related one. Yeah, no, my advisor would be that's his one of his favorite ones. So yeah.
Yeah, they do
And
call them midges. They use the same common name for
yeah,
Forsythia and Culicoides, so everybody gets confused.
it's the sister genus. So
Okay, so
yeah, yeah.
so the general, so okay, but you were basically saying I don't work with so these are ceropotogonids. So
Yes, yeah. It's ceratopogonids are the reason you have chocolate. So you're welcome. But
exactly. So that was what I thought was sort of interesting. So one question I had about the use of these. So this idea, the original idea of actually using the trapping as a way to reduce the fly
yeah.
population, it would be like a twofer, right? So,
Yes.
but why not use more? If one doesn't work, then try again, right? So,
Yeah, no, we're continuing to do that, right? And actually we're on a new sort of iteration. The mass trap in its initial, so Lee's initial thoughts were to use it as mosquito surveillance. And so we've sort of diverged. He's continued to use it in the mosquito surveillance world, and potentially it could put, you can collect enough biomass of mosquitoes to do what we're talking about doing. But when I came in, I was like, we should do house flies. And so we've sort of split the work, and he's continuing to do surveillance and testing for viruses that he's collecting with the mass trap, and then also using them for biomass. And I've gone straight biomass with the house flies. When we collected those 12 kilograms, that was using a couple traps actually instead of the singular one. But that fan, the amount of flies that we have, that fan actually clogs up, and so we're working on a new design that potentially utilizes a different mechanism to bring the flies in. But I think you're right in that you just put out more traps until you hit to where you're hitting, reducing the population.
it's because the trapping is definitely an approach that has been now developing in the mosquito world for actual control.
Yes.
The way you sounded, it's like, well, would be bad because then there would be not enough food, but then there would be less flies. And
yeah, no. Like if we could do both, like that would be the best, right? And so, but the way that this facility is set up, it's an ag facility that's producing its own houseflies surrounded by nothing but corn and soybean fields that they then take the manure and spread on those fields as well. So, I mean, I pulled like 2 million flies away from them in like 8 weeks, and I didn't dent the population. I never saw a reduction in the population. So, yeah.
got it. All right. Now I'm just
I mean,
really
at some point we can remove as many flies as they want, but if they're hell bent on producing them, there's nothing we can do about it at the, like that IPM level. So
I also find, and the fact that it was done for dog food, it was confusing to me, because I would have thought that'd be something like a chicken feed, like a farmer has chicken,
yeah.
like you said, you had poultry, you had things, so that would make more sense. So, but somehow The dog.
Yeah, I mean that's Purina's, right? There's just a commercially available dog food that has feed in it already, or insects in it already. So
Okay.
yeah.
Shut up. Grayson has a.
Hi, Philip.
Hey, what's up?
Not much. I think you sort of like a kind of addressed something I was going to ask about in that you said you didn't see any like dent in the population even when you caught a ton of flies. But I guess if you did, would you anticipate that there would be any negative effects of like removing a large amount of insects from their ecosystem?
Yeah, so this is definitely something that we considered, and why we targeted the livestock facilities, because those livestock facilities are producing those sort of unnatural amounts of insects. And when we subsampled our collection, we found that less than 1% was anything other than a filth fly. And so, you get into a debate of like, if we artificially create an ecosystem that has 7 million houseflies in it, that maybe these possums that are around and depend on, or the spiders that are in this, they're depending on this sort of artificial population that we've created. Is it wrong of us to then take that away from them? And I don't really have the answer, but I definitely think that it would be not good to use this type of trap in sort of a more natural setting, because you would get absolutely way more than just the flies. But it is something that we took into account, and we subsampled what we were collecting just to make sure. I did absolutely kill a ladybug. I'm so sorry.
Yeah, no, I mean, thank you. I was thinking about, I forgot the exact details of his story, but something about treating cattle and it ended up having an effect on like scarab beetles and their decomposers. So it just like ended up that manure was piling up, which is like a different thing. But yeah, thanks. Great talk.
Yeah.
Really interesting.
No, I mean, that's true too. If like, because we are taking like the houseflies, it's not just a food thing. Like the larvae are actually doing something in the environment, and the reason that they get those high populations is because they're decomposing and things like that. So if we take that away, yeah, potentially you could have other problems. So no, it's definitely something to think about.
Okay, that made me think of another question.
Yeah.
Okay,
Alex, do you want to stick around with the class afterwards?
Yeah, I'm fine. This is my last question.
Okay.
Yeah, wait, what was it? Oh god, now I went blank. Oh yeah. What kind of diet are the animals on these facilities eating? Like, what sort of diets are they being fed?
It's definitely corn based.
Yeah, that checks out. That's why there's probably so much manure. Why there's such a natural number of flies.
Yeah.
Probably it's the lack of omega-3 in their diet that's causing that. Like,
well, and the flies love the corn too. So as it gets milled in the silo, it gets milled and then put in and fed into the coops and things like that. But there's some of that dust gets and accumulates on the ground, and the flies are all over it. The maggots are all in it.
And that combined with all the manure. Oh, wow.
Oh yeah.
Okay.
Yep. No, it's a wonderful place. And
All right. Thank you very much. That was a lot of fun, Phil. So for the folks in the class can stay on the link. Everyone else can sign off. Maybe what we'll do is just take a few minutes, take a quick 5-minute break, and then we'll meet back here. Everybody turn on your cameras. I'll have Steph stick around as well, because I have to bail at 12:50, but I have a bunch of Midge questions I'll